but I wanted to talk about today some of the work I've been doing since I started in 2010. I work in an endocrinology-focused lab, and our overall goal is this idea of predicting phenotypic characteristics in animals. It can even apply to the human side, and I'll get into that a little bit more as we go along. And so the title of my talk is, what I'm basically saying is I want to develop a way to measure many hormones at the same time, because we can't do that right now. And so I start off this talk with this next little graph of human blood glucose. And so the point I want to make with this is that even if you're not a trained scientist or an endocrinologist, you know a little bit about glucose, where you can probably make some predictions of what's going on here throughout the course of the day. And if you were to do that, I'm sure you'd come up with this. Each of these peaks in glucose is probably a meal that this person had throughout the day. And so probably had a big breakfast, a medium lunch, big dinner, and then a snack before bed. Now, where the endocrinology comes in is that I can make assumptions or predictions of other hormones based on this profile alone. And you might know this as well, but I can predict insulin levels in the blood as they relate to the glucose peaks that we see. Because I know that after a meal and after a glucose spike, an insulin spike follows not long after it. And so what I'm trying to say here is that we can make inferences or predictions based on a single profile over some amount of time. And so to give you a more relevant example as to what I work on is cattle. And so if I'm somebody who's interested in getting some money for these beef cattle, if I were to look at these two in the field, I'd have no way of telling which one of these is a high-quality beef steak, which actually, I'm not sure if you know this, but this is the higher-quality beef steak versus this one. I can't look at these cattle and know the difference and know which one of these is going to bring me this higher-quality beef. And if we break that down into numbers, that's a net profit or loss of $156 per animal. And for the most part, cattle operations are fairly large, so that's going to add up over time. And so some previous work in our lab has actually determined that a relationship between marbling score and serum hormones, in this particular case leptin, which is a hormone that's actually secreted by fat tissue, but they've established this relationship that exists between the amount of marbling, or the amount of fat, and the level of leptin. So that makes sense, because leptin is secreted by fat tissue. So they saw this data, and they said, well, what if we expanded that and we tested 3 different hormones that are involved in muscle growth or muscle production as well as fat? So this data set represents 30,000 animals, 30,000 cattle, with a single blood sample that was collected and slaughtered. And what you can see is that with each hormone, a very distinct relationship exists. So with USDA quality grade, and these are all just measures that a person at the slaughter plant gives the steak a certain grade, a certain yield grade, and then marbling scores that ultimately determines the price. And so they established that a relationship exists between the levels of leptin and quality grade, IGF, or insulin-like growth factor, and yield grade, and growth hormone and yield grade. And so basically, as this decreases, you get an increase in yield grade, because that's the amount of fat. So that's like the fat to muscle ratio. So growth hormone and IGF are both involved in muscle growth. And so as our levels of growth hormone decrease, then we have more fat in that steak than muscle. And so after they took those 30,000 animals, they created this algorithm that says if I plug in this animal's hormone value, I can come up with a number that correlates to, so if I plug in so they took 1,700 animals and they plugged in those hormone values into their algorithm that they created in order to predict how that USDA grader was going to grade those animals. And really the point here is that the misclassification rate, or the rate at which they got it wrong, was less than 5% throughout 1,700 animals. And so this is prior to slaughter, before the USDA guy even sees the carcass. And so they're already saying, even before the slaughter, they can predict how those animals are going to grade out, and consequently, how much money that's going to bring. So my advisor wanted me to put in a little shock value here. This is all important because of the speed of commerce, and I'm going to draw your attention to the number of animals we process per minute and per day. So right now, in 1 minute that's passed since I started talking, 63 cattle have been slaughtered for meat production. And by the end of today, 90,000 animals will be slaughtered. And that's cattle, and obviously the number jumps for chicken. Point being is we're slaughtering a lot of animals, so if we wanted to do this prediction thing, it would take up a lot of time, because each one of those assays takes about 3 days apiece. So that's a lot of animals with a lot of very large assay, and we're very restricted in our assay flexibility, and I'll get into that a little bit more. So if I were to make an analogy of running these assays, it'd be something like this, where each one of these cells is represented one of those 90,000 animals. And so that's a lot of bullets that goes into reaching this hormone of interest. So if actually goes into determining that algorithm just grows exponentially. And so it's kind of a hindrance. Now it's really cool that we can predict this, but there's still a number of problems. But to summarize what I've said up to this point is that we can in fact predict carcass quality with a single hormone. And we can improve that predictive capability by measuring a panel of hormones from a single blood sample. The hormones that were used in the previous data included these growth hormones associated with muscle growth and fat deposition. And then the hormone prediction matched the actual classification results 95% of the time. So the algorithm is good. It works. But there's still a number of questions that we have not answered at this point. Can we add more hormones to this panel to increase our predictive capabilities even more? So we saw kind of an additive process with the 3. Can we expand it out to even more hormones? Are there other phenotypes or other characteristics that we can predict that are important in animal production, including growth rate and reproduction? Both of these are also very lowly heritable on the genetic side, which is this is kind of separate from genetics, and so we want to address some of these potential issues. And then my research is focusing on how can we make this profiling more efficient, because like I said, with each one of those hormones that you want to analyze, it takes at least 3 days, and you can really only run about 10,000 samples a day, which is not good. And so the current detection methods that we use in the lab are antibody-based detection methods, and we include the radioimmunoassay, which was first developed in the 1950s, and the enzyme-linked immunoassay, which was first developed in the 60s. The basic premise for all of these is you have an antibody of interest to your hormone, and it binds your hormone of interest. I'm going to use the radioimmunoassay as the example because that's what we use in our lab currently. And so what we do in this assay is we add a fixed amount of a radioactive antigen. So that's another potential problem. Radioactivity, a lot of people don't like it. They're scared of it. It's very cumbersome to work with because you have to get all these, you have to be certified by the NRC and such and such. But what we do is that we add a fixed amount of our labeled antibody, or labeled hormone, and then we compete it with our unknown sample for a predetermined amount of time after that 3 days comes in. And then so there's some kind of binding, the antibody is going to bind the radio-labeled and the non-labeled in a specific manner. And essentially we can measure, so the reason we use radioactive is so we can trace it, so then we put it in a machine and we read it, and we can determine from that the amount of displacement or the ratio of the labeled to unlabeled can give us a quantitative measurable value of our hormone. So as I've kind of alluded to already, each of these radioimmunoassay and the ELISA have a number of limitations. They tend to be expensive. They may or may not be sensitive or specific enough for the hormones that we're interested in looking at. They take a large amount of time. And then again, the fact that we can only measure one hormone with one assay. So we need 3 separate assays for 3 separate hormones. So I'm looking to address this issue with multiplexing, in which one sample can be used to measure multiple analytes. This is important for like model species like rodents and rabbits because you can't get a lot of sample out of them in the first place. So if we can multiplex, then we can save some of that precious sample that gets used really quickly, and it increases throughput, which is probably relevant to today's, the symposium today. It also has the high potential for repeatability because most of these technologies are actually based in a bead format instead of a solid matrix, and so each of those beads can count as a replicate or a redundancy in our sample set, which is really great because at the most we can only do about 3 samples, 3 replicates in the radioimmunoassay. So there are currently 2 platforms that I'm aware of that utilize and work on multiplexing, and they include the Luminex platform, which is these really tiny microspheres that are made of polystyrene. They use polystyrene because what they do is they dip it in a proprietary dye, and then this ratio of dyes creates a unique signature for each set of beads. And so they make 100 different bead sets, which means I could multiplex up to 100 different combinations of hormones in my sample. There's also the Illumina BeadXpress platform, which I'm sure you've all heard of Illumina because they do the genotyping stuff. And what they use is they use these glass beads that are about 20 little tiny micron rods. Each bead is actually etched on the inside with a unique barcode, which is pretty fascinating. And they have up to 600 combinations available, very similar. I could mix any set of barcodes I wanted and label them differently with my hormones of interest. And then others that I may or may not be aware about, I don't know. So my overreaching hypothesis throughout my research has been that we can optimize the existing multiplex technology for hormonal analysis in a multiplex format. Surprisingly, it's not been done. They've done it with cytokines for inflammatory and disease purposes, but not really for this predicting, which is what we're looking at. And I'm not really going to show any of my data because it's nothing terribly exciting. Most of it was trying to validate these different platforms. But again, I wanted to give you guys like the story of how I'm using this technology. And so I started out in 2010 on the Illumina BeadXpress platform. As I kind of explained already, there are these glass beads that have a covalent modification that allows you to stick a protein on them. And an antibody is a protein. So we can easily stick our antibodies onto our bead surface and then incubate them with our sample to have them stick. And then we come in with a fluorescent reporter utilizing the science and strip-down interaction. All you really need to know is that's a really strong bond. And it's very, we use it, it's almost as strong as a covalent bond, if I remember correctly. And so basically, it links it pretty tightly, so it's not going to fall off. And so then you incubate it with a fluorescent recorder because inside this machine there's a couple of lasers. One laser reads the barcode on the glass bead, and the other one reads the fluorescence that comes off the bead, which should be relative to the amount of sample that I've added onto my glass bead. So the more sample I have, in theory, I should have more fluorescence. And so the Illumina BeadXpress platform was actually originally used for genotyping assays. It was one of their first platforms they released, and then they kind of opened it up for these protein-based assays by doing that covalent modification. And this is what it looks like in the lab, and this is what they look like under a microscope. Pretty cool. So there are a number of challenges that we had when we did this work with the Illumina and some lessons we learned. So one of our biggest challenges is we are not biochemists, I'm an animal scientist. So there was so much biochemistry, it was so frustrating. I could not wrap my head around a lot of it that was happening. And so that was a huge challenge, and I spent most of this first year of my PhD trying to figure out how to make the antibodies stick to the bead. It was not as easy as they said it was in the literature. Another thing that we didn't really anticipate was how are we going to remove the liquid from these assays, because everything that we combine with is in a liquid suspension. But you don't want all the extra stuff in there. You want to add more than what you need, and so whatever sticks is going to stick, and then you wash off everything else. And so the problem we had was, well, how are we going to wash everything off without losing those beads? So we devised this really elegant and cheap aspiration device that's essentially a gas chromatography column stuck into this metal hole so that it cuts back in the vacuum so I don't suck up my beads, but I can remove the liquid pretty easily. Another thing is data analysis, which I've talked to a number of people about today, because each one of those beads in like that previous picture is its own data point. And so if I have if I have 100 replicates or 1,000 replicates based on the number of beads, how are we going to handle that giant increase in data? So we didn't really know how we were going to approach this. And then the other question was how much data is enough? Is one bead enough, or do we need 100, or do we need 1,000? And those are some of the questions we had to think about going into this. And then ultimately, number one challenge is last summer they came to us and said we're no longer supporting the machine. So I was pretty much, I had to go pick a few things after that. And so some of the lessons we learned was, don't believe everything you see at face value, because there's a chance that it's not going to work the way the company says it is. And I don't want to bash the company. They were extremely helpful. It was just really difficult. And they were limited in their knowledge of how things worked the way that I needed them to work. And so it made it a little bit tougher to move forward. And then we also figured out that no matter the format, aspiration is going to be an issue. So luckily we devised this little apparatus. So it became like, yeah, look at us, we did it. So after the unfortunate demise of the BeadXpress platform, I went ahead and moved to the xMAP technology, which I mentioned already. They're these polystyrene microspheres. And so it's actually very similar to Illumina. It's just that their bead differentiation is different in that the beads fluoresce internally, and that's how they determine bead A from bead B and so on and so forth. And then they use the red laser to excite the fluorescence. So everything was the same, just except what the beads were made of and ultimately how it processed them in the machine. So the same thing, we had a number of challenges and lessons we learned from the Luminex. Primarily, the polystyrene consideration was something we weren't expecting. That kind of goes back to biochemistry again. There were some considerations for the matrix that we were working with, and then like putting them in the plate, and then the chemistry of the cross-linking, all issues we didn't even consider. Again, aspiration, and then limited flexibility, which is something that's really important that I'm going to get to here in a second. We also learned at this point, we got a better understanding of how biochemical cross-linking works, and I read books, and I think I've kind of figured it out now. We moved more forward a lot faster this time than we did previously. And then we started to learn how to handle those large data sets, and we're kind of getting an idea in our mind of how we're going to handle that. Of course, I'm open for input on how to handle those. So at this point, I wanted to make sure that you guys are understanding that we're still using this old technology to measure our samples today. And as we have this increasing need for demand of information, it's not going to cut it. We need something that gives us more information faster. And the bead-based platform may actually fill this need. We need less sample, so we can use that sample more wisely, and then we can meet this demand for information through the multiplexing format. So at this point, we're at a crossroads. Do we continue to develop the assays on the Luminex platform, which were advantageous because they worked, and it was like, yes, we're making progress. But the problem here that we had was the restricted flexibility, and what I mean in that is that we would have to be 100% reliant on the company for consumable reagents, including the bead sets. We could not make them ourselves. And the machine, we tried. We met with some chemists and tried to make some of our own, but the machine is also very tightly regulated that runs the beads, and so we didn't have any kind of flexibility. We would be tied into that company forever. And after what happened with Illumina, we were very cautious about that. So our alternative was to develop an assay that wasn't reliant on that proprietary technology. Now the hesitation is that there's no guarantee it's going to work, and it kind of brings me back to square one again. But we have improved flexibility because we don't have to rely on a company. We wanted to build something that didn't require any kind of proprietary technology outside of maybe some basic machines that we could use. So ultimately, this is the route we decided to take, and what we decided to develop was this Immuno-PCR. And in talking to some people earlier, I understand that PCR is probably not a term that most people understand in this kind of setting, because I'm used to that. It's a very widely used technology in molecular biology that essentially takes a small piece of DNA and copies it over and over and over and over again. And so you amplify a little piece of DNA. And so they use it in like forensic sciences. If you want to try to figure out who done it, they take a little piece of that DNA, and if there's enough there, they can amplify it and get a sizable amount that they can start manipulating. And so we're kind of taking that standard technology and changing it around a little. It's been described, and it was first described in 1992. And in this case, we're using antibodies and DNA to detect our hormones. And so the antibodies, again, are used to grab that hormone of interest. But instead of using a fluorescent recorder that relied on the machine that has a specific laser, we instead use DNA as a recorder. So essentially, it's doing the same thing, but we're not using it to do a whodunit. We just wanted to say, this antibody is attached to this piece of DNA, and so it makes that many copies. And so we can then synthesize any of our DNA of any size we want, change a base pair. You know, we can do whatever we can. I mean, these companies, they make DNA for you. I want this sequence made, they'll send it to me. So all we have to do is come in and make that, and you know, that's like all the flexibility you need. The other great thing about PCR is it's incredibly sensitive. And so in my early work in this foray, I can detect over 4, 5, 6 orders of magnitude of DNA, which can then relate back to my protein. So I can detect minute levels of protein that even my conventional assays cannot detect. And so I've kind of talked about the advantages already. It's sensitive. It has the flexibility. Nothing's proprietary other than the use of antibodies, which most companies make antibodies. So if you want something you want to detect, there's probably an antibody for it. Also, customization is very interesting to us. We can set up an assay in a number of different ways and still potentially get the results that we want. And so this is just an example of how Immuno-PCR has progressed since the initial description in 1992. And as you can see, you could run with any of these different types. I'm not going to go into too much detail. But any sort of these configurations can work to our advantage. Some of the challenges include those assay designs with the double-edged sword, because what if one works and one doesn't? I'm going to have to test all of those before I know that for sure. Will it also, I mean, does it, that whole we don't know if it's going to work thing. Of course, the published data says it should work, and I've done some early preliminary testings and it should work, but it's still too soon to know for sure. And I mean, it wouldn't be science if we didn't try it. So I'm going to go headstrong into this either way, but it's, you know, it's a little bit nerve-wracking to have it. What if it doesn't work? And then something else that we're interested in is improving the design of the Immuno-PCR, is taking it that one step further. So as it exists right now, what they actually do is they create like this lawn of antibodies and they stick them to the bottom of the plate. And so basically you have a bunch of hands, like if you think of like zombies coming out of the ground, they're all grabbing for something, but they're stuck there. And so what we are trying to do is actually marriage the bead-based technology that I discussed earlier with the Immuno-PCR by actually throwing those beads in there and then having them be this, you know, they can actually churn in the reaction and you have more surface area than you would versus a plate. And so what we want to do is try to use the bead surface as the matrix for the antibody to bind to maybe potentially increase sensitivity even more magnitudes. It'd be really great if this works. I have actually, so when you do a PCR, you basically put this in this machine that gets hot and then gets cold, and hot and cold, hot and cold. So we call it a thermal cycler. Pretty straightforward. And I put my beads that I want to use for this into the thermal cycler and tried to run a PCR, and I've had no interference. So the early work is actually really promising that this is going to work. And that's going to be really cool because it's taking that technology to the next step that we think would be awesome. And then, of course, any other potential pitfalls that I haven't considered yet. Of course, data, again, is going to be an issue. And this one is a little bit more tougher because we are very limited in our molecular biology experience. So learning how to handle that kind of data is going to be a challenge, but I don't foresee it being a problem. So my final summary is that hormone multiplexing is an essential tool for predicting phenotypes. And phenotypes outside of animal science. What if we could predict if you're susceptible to disease, or if we can, you know, tell you if you're, you know, I guess disease is the number one, and any other kind of endocrinological things that could happen to a person. The multiplexing formats are varied. Illumina BeadXpress is unfortunately being phased out. The xMAP is useful, but it's limited in its flexibility. And the Immuno-PCR at this point appears to be both flexible and sensitive. And using multiplexing should increase our capacity for phenotype prediction, and then looking to these other areas of animal production and maybe even disease prediction, which are another very important facet of animal production that we don't really have a prediction for at this point. And so what I can plan on doing when I get back to the zoo is just continue developing this technology using the Immuno-PCR. And with that, I will acknowledge all the people that have helped me. Dr. Dwayne Kiesler is my advisor PI. He's the one that came up with all these ideas. Very awesome person, and we love technology. And then my committee members, and then other various people that have helped me in the laboratory. And then I'll take any questions. And what this is how the Illumina BeadXpress platform actually worked. When I first gave this presentation, and it took me a long time to make this animation, so I left it. And so that's kind of a visual of how the fluorescent bead assays work. They're very similar. Thank you for coming.